Hello students, so in continuation to lecture number 3, here in this class we will study about the cloning vector in plants and animal, and we also discuss another topic, that is competent host, one by one. Okay, first come to cloning vector for plants. So when we come to plants, most of the dicot plants are infected by a particular bacteria by the name Agrobacterium tumefaciens, and this particular bacteria, it induces the formation of cancerous growth, which is known as crown gall tumor. And this transformation or disease is induced by a gene, and this gene is present in the plasmid of Agrobacterium tumefaciens. That's why this plasmid is known as tumor inducing plasmid. Okay, now this TI plasmid can be made non-virulent by deleting or by cutting the gene or part of the gene, okay, part of the DNA which is responsible for the tumor, and the part of the plasmid which is transferred to the plant cell DNA is known as tDNA. So the point is that if the desired gene is able to insert into this tDNA, then the modified plasmid can still infect the plant, but there will be no tumor. This tumor will not be produced. So what is the point? We can now use this TI plasmid of Agrobacterium tumefaciens after deleting the gene responsible for the disease, and it can be used as vector to transfer the desired gene into the plant. Okay, aduna sida harise kaina kanda bacterium biotechn Agrobacterium tumefaciens haisina plants ta kadoke kanda crown gall tumor haba hogi cancerous growth ama induce tawe. Ado hariba sigi cancerous growth se induce oriba aduda responsible toriba gene dudi krai yauge kanda plasmid yawe kanai plasmid ta Agrobacterium tumefaciens gi aduna ngasagi plasmid se akoina kari kaure tumor induce tawe na kayuna TI plasmid kaureba aduna sigi TI plasmid se akoina kadoge kanda ngasagi lena thoram jin do lawthuoga non violent ohenba yagani habane aduga sigi TI plasmid the particular DNA material known as tDNA sigi tDNA sneba ngasagi aduga host ki cell ki DNA da transfer to gadai adui taragadi tDNA gi hariba jaga sida akoina apamba gene do hapsin ngamba taragadi ngasai ga dodiba akoigi modify toroba plasmid tuna hayeng akoina animal ki sorry plant ki cells da infection tor matam da magi infecting process to jaba taraga su there be no disease havene kaina kanda disease one of gene do ko thokrabani na sumai to dana TI plasmid of Agrobacterium tumefaciens se plants di vector oina sinaba yagani have. Okay, next come to cloning vector for animals. We know that animal including we human being are infected by retrovirus. So here what can we do? And this retrovirus can be made disarmed. That means we can cut the gene responsible for the disease, and in that particular region you can insert the desired DNA, and it can be used as vector to transfer the desired gene into animal cell. Aduna akoina kadoba kya kanda akoi animal sidi retrovirus na infect tobunina retrovirus gi gene do karam gene no yakanda ngasagi infection thonam gene do lokhoga aduda apam gene do hapsalaga ngasagi infecting process to anba yagani haneba or sigi process to matabna kadogani yakanda ngasagi apam gene do introduce to gane. That means retrovirus can be used as vector, okay, in case of animal. Aduga plants na we have to use TI plasmid of Agrobacterium tumefaciens. So this is about the cloning vector for gene in plants and animals. Next come to competent host. Here the recombinant DNA which was produced during recombinant DNA technology, it cannot be directly introduced into the host cell. This is because we know that this DNA, it is a hydrophilic molecule, and on other side every cell is surrounded by plasma membrane which is made up of lipid bilayer. Because of this, the cell cannot be directly into the host cell. So here what we have to do is we have to treat the host cell to make it competent. So here to make competent means we have to make the host cell capable to take up recombinant DNA. So to make the host cell competent, the host cell should be treated with a specific concentration of divalent cation, just like calcium, and this process will help to increase the efficiency with which the DNA will enter the bacterium, okay, through the pores. After this, the recombinant DNA can be forced into such cell after incubating the cell with recombinant DNA on ice, and it should be followed by heat treatment, okay, around at 42 degrees Celsius, and again it should be followed by cold treatment by putting them back on ice, okay, and after this the foreign DNA can pass through plasma membrane and it can enter the cell. Like this, the host cell will be able to take up the recombinant DNA. नंगासागी recombinant DNA से host substandringa host cell से हाना competent और चंगी है करीगिन है काना DNA है बसे हाइड्रोफिलिक मॉलिक्यूल ने और सेल दी कनाने क्रॉस अभी प्लाज्मा मेम्ब्रेन कनान सेमी लिपिड बाइलेयर अदुना DNA दुदी नसागी सेल मेम्ब्रेन फॉर समंगा मोय अदुई तारदी होस्ट ना recombinant DNA तो एक्सेप्ट होंगे है तारदी होस्ट से होना competent और हम चंगे हाइबदी होस्ट ना recombinant DNA तो एक्सेप्ट होगा लौसमंगा हम चंगे अदुना लाइदी क्वांटो ट्रीटमेंट तोगनी है अदुना होस्ट सेल से अखोइना कदोगनी है कांदा अखोइना केमिकल हान ट्रीट तोगनी मरुइना कैल्शियम दा सिना कदोगे है कांदा ngasai कदोबा मागी pores फॉरगा DNA चंपे सांग से हेलनी आफ्टर दिस अखोइना कोल्ड ट्रीटमेंट हाई कई तौदेनो ngasai केमिकली ट्रीट तौरबा होस्ट सेल सिने recombinant DNA सिने कोल्ड ट्रीटमेंट पिगनी फॉलोड बाय हीट ट्रीटमेंट है कांसदा when hit together around 42 degrees Celsius da again moi ka dawni amu ice thamgani amu ka dawni cold treatment pigani adu kanda ka dawni ha kanda ngasai ki recombinant DNA do plasma membrane phawraga changaga ngasai ki cell samanga mane smain to dana akoina apamba DNA do introduce obu yani smain to dana ngasai ki host cell ka dore host cells wina competent ho hemba yagane so this is how we make the host cell competent. Next come to direct transfer. So now you can transfer this recombinant DNA into the host organism without making the host cell competent, and this can be performed through microinjection or through biologic origin gun or through the use of disarmed pathogen. First come to microinjection. So in this method or in this procedure, the recombinant DNA can be directly injected into the nucleus of the animal cell, and this can be injected with the help of micropipette, and this method is mainly used in oocyte, egg and embryo. Next come to biologic origin gun. In this method we have to use a device which possesses microscopic pellet, and this pellet may be made up of tungsten or gold particle. And in this method what we are going to do is that the desired gene or desired DNA will be coated onto the microscopic pellet, and then it will be shot into the target cell. And this method is mainly developed for plants, but still it can be used to deliver or to insert the desired gene into animal cell also. So this is about biologic origin gun. The last method is the use of disarmed pathogen. Here we can use the pathogen as vector by deleting those genes which are responsible for infection, and in the region where we can insert the desired gene, and we can make this pathogen as vector to transfer the recombinant DNA into the host organism. So this is another way to transfer the recombinant DNA into the host organism without making the host organism competent. Aduna hawjiti recombinant DNA se host cell absan yare without making the host cell competent. See the way lambi ahum lai habani. One is microinjection, one is biologic origin gun, one is use of disarmed pathogen. Microinjection to this se kemya kanda koina micropipette haiba device am sinaraga recombinant DNA do ngasai gi cell ki nucleus da cell DNA of nucleus da direct inject option ga dene. Biologic origin gun da koina device am sinagani sigi device da pellet am yawi sigi pellet se mai se gold nataga tungsten semgan habani sista apamba gene do hapsalaga direct nomai kapadona target cell so direct option ga dene. Like that akoina pathogen se disarm toraga mase bheitar insina yagane so disarm to bhaitaraga di leina thona gene do lothoga apam gene do hapsan dai aduga pathogen sing do akoina kadore bheitar insina yagane. So these are the three ways by which we can introduce the recombinant DNA directly into the host cell without making the host cell competent. And this brings me to the end of this class, and today in this class we have discussed about the vector for cloning gene in plants and animal, about competent host, and about how to transfer the recombinant DNA into the host without making the host cell competent, and hope you can understand each and every point. And thank you very much for your attention.